I would like to thank the organizers of Canola Week for the opportunity to share our exciting results with you today. And I've entitled today's talk, Nutrigenetics, Canola Oil, and Blood Glucose Levels. Does SCD1 genotype modulate a person's response to canola oil? And before I begin, I would just like to disclose that I have received funding from the Canola Council of Canada, and that's actually the funding that supported the work I'm presenting today, as well as from agencies like the Dairy Farmers of Canada, OMAFRA, NSERC, and CIHR. I'm also remunerated for my role as editor-in-chief of Lifestyle Genomics. So when it comes to understanding the model that's currently used to make dietary recommendations, we need to recognize that those recommendations are made at the level of populations, and therefore we use population averages to guide the development of these dietary recommendations. However, they do not take into account the individual variability in response to nutrients. In other words, the fact that different people will respond differently to the same nutrient. And so the current model that's used to develop dietary recommendations uses something as simple as this, is that with the consumption of a nutrient, it will have some effect on a health outcome of interest. And that effect could be positive, it could be a neutral effect, or it could be a negative effect. But this is the model that's being used. But the reality is that we are all different. So it stands to reason that our responses to these nutrients could vary from one person to another. And in fact, this is best demonstrated and exemplified by some of the work that we've done following the consumption of canola oil. So there's a study that was conducted and led by researchers at the University of Manitoba called the Comet 2 trial. And this was a trial that looked at different types of oils that were incorporated into the diet. And so I'm focusing here just simply on what happened in individuals following the consumption of regular canola oil in this particular study. And after 6 weeks of consuming regular canola oil, we were interested to look at what happens in blood glucose levels. And this was in about 106 individuals consisting of both men and women. And so what we know is that blood glucose levels have been reported to decrease with the consumption of canola oil. And sure enough, if we look at the average of this 106 individuals in terms of their change in blood glucose levels following the 6-week consumption of canola oil, we can see by looking at this dotted line here that there is a slight reduction in blood glucose levels after consuming the regular canola oil. However, the reality is that everybody responded very differently to the same diet. And in fact, if you look at the individual responses, where each blue bar represents one of the 106 individuals in this study, what you can quickly see is we can actually divide out our responses into positive responders, individuals who experienced a strong reduction in blood glucose in response to the consumption of canola oil. But we also had individuals who had very little response, neither an increase nor a decrease. It just basically didn't change. And then we had a group of individuals who showed an adverse response. In other words, their blood glucose levels increased. And so it's very clear by looking at data like this that individuals will respond differently to the same foods. So therefore, the population-based recommendations can definitely be improved upon. And so if we try to understand then what are the different factors that influence and underlie this individual variability in response to diet, again, we have our simple model here of nutrient to health outcome. We have to recognize that there's a lot of different factors that come into play here. So for example, lifestyle factors, how a person, the dietary habits, the exercise habits, whether they smoke, whether they consume pharmaceutical medications. Those types of factors can influence the gut bacteria, which influences host metabolism. And at the same time, we also have to think about at the systems biology level, the biochemical or genetic factors that can influence and that can interact with these lifestyle factors to also influence how a nutrient affects a health outcome. And so we can look at things like genes, proteins, and metabolites and how these different factors interact with one another to create these sort of complex networks. So it very quickly becomes very, very complex and very challenging to try to tease apart and identify the underlying factors that control a person's response to a particular diet. Now, my lab is very interested in the field of nutrigenetics. And nutrigenetics is, at its simplest, how do genetic variants or genetic single nucleotide polymorphisms in the genome, how do they affect response to a nutrient or a diet? In other words, we know genetics will vary from person to person. What is their role in terms of mediating that response to a particular diet or nutrient? And so if we think about the Nutrigenetics model, we have to actually add in a layer, further layer to that simple model that I presented initially, how a nutrient influences a health outcome. And so what we have to recognize is that nutrients will interact with genetic variants in the body. And those genetic variants can then subsequently have a very different outcome or a very different association with that health outcome of interest. So you can imagine that we have different genotypes that exist at that one particular genetic variant of interest, genotype A, B, and C. And if we looked at individuals and separated them based on their genotype, we could actually get much clearer information about how that nutrient influences the health outcome, because we could see that individuals with genotype A show an improvement in that health outcome, individuals with genotype B show no effect, and individuals with genotype C show a worsening in the health outcome. So essentially what this nutrigenetics model really emphasizes is that the one-size approach that's used with population-based recommendations is not appropriate for the individual. In other words, one size does not fit all. So the goal of our proof of principle secondary nutrigenetics analysis was quite straightforward, is we were interested to examine if the effects of canola oil on blood glucose levels were influenced by single nucleotide polymorphisms, or SNPs for short, in the SCD1 gene. And I'll explain what SCD1 does in a subsequent slide. But we hypothesized that people with a low conversion efficiency SCD1 genotype would experience greater improvements in fasting blood glucose levels following the consumption of canola oil compared to individuals with the high conversion efficiency SCD1 genotype. So what exactly is SCD1 and what is known about SNPs and the association between SCD1 and insulin sensitivity? Well, SCD1 is an enzyme that's known as sterol coenzyme A desaturase 1. And essentially this enzyme is responsible for a very specific biochemical conversion of fatty acids in the body. As you can see here, it converts saturated fatty acids, or SFA for short, into monounsaturated fatty acids, or MUFA for short. And I'm just demonstrating over here on the right-hand side that a saturated fatty acid like stearic acid is converted into oleic acid thanks to SCD1. And what SCD1 does is it biochemically introduces a double bond into the fatty acid structure, creating this sort of kinked structure. Now, monounsaturated fatty acids, or MUFA for short, are associated with improvements in blood glucose levels and whole body insulin sensitivity. Now, why we were particularly interested then in SCD1 is that it's known that there are SNPs in SCD1 that alter enzyme activity. In other words, you can have an SCD1 low conversion efficiency or a low conversion genotype, where the genetic variant that exists in SCD1 reduces the conversion of saturated fats into monounsaturated fats. But there's also then another type of genotype where you have a high conversion efficiency. In other words, there's a form of SCD1 that increases the conversion of saturated fats into monounsaturated fats. So using the individuals that were recruited in the Comet 2 trial. So the Comet 2 trial was a fantastic way for us to explore this nutrigenetics question in both men and women who had abdominal obesity. And as you can see here, participants consumed 3 treatment oils in this randomized double-blind crossover study. And the treatment oils consisted of a regular canola oil, a high oleic acid canola oil, in other words, a high MUFA content, and then a saturated fatty acid enriched control oil. And these oils were incorporated into a smoothie beverage and consumed as part of a full feeding diet. Each oil was consumed by each person for 6 weeks, and there was a washout period of 6 weeks between each of the oil consumption phases. We used data on fasted glucose and insulin that was collected before and after each treatment oil, as well as samples of DNA for genetic testing in SCD1. Now, our primary finding that I'm going to just report on today was, first and foremost, that we did not see a general effect of canola oils on blood glucose levels compared to the saturated fatty acid enriched control oil. In other words, the changes that were experienced in blood glucose levels with the canola oils compared to the saturated fat enriched control oil, they weren't significantly different from one another. However, when we stratified individuals based on their SCD1 genotype, what we found was that individuals who carried the CC genotype, which has been associated with a low conversion efficiency, in other words, their body isn't as good at making MUFA as individuals who carry the AA or AC genotypes, we found that these individuals, following the consumption of that control saturated fatty acid control oil, they showed an increase in their blood glucose levels. But however, when they consumed the regular canola oil or the high oleic acid canola oil, you can see that they showed reductions in their blood glucose levels. And this was shown both in an additive model and in a recessive model. So this highlighted that the genetic variant that existed in SCD1 modifies blood glucose response to dietary oils in individuals who carry a very specific genotype, the CC genotype. Now, if you're interested to get more information about this study, I would point you towards the publication that was published earlier this year in 2021 in the British Journal of Nutrition, and as well as an infographic that was generated by my students that describes very simply the relationship between canola oil, blood sugar, and your genes. So to conclude, we identified a SNP in the SCD1 gene that modified blood glucose response to treatment oils. Therefore, individuals carrying this rare CC genotype in the RS3071 SNP could benefit from using a canola oil in their diets compared to a saturated fatty acid enriched oil. Now, this SNP could potentially and eventually be used by healthcare professionals who are using a personalized nutrition approach using genetics to manage blood glucose levels in their patients. However, we would need to consider more research in this area to validate the use of this SNP as a tool to not only motivate dietary behavioral changes regarding canola oil, but also its use and its application to personalized nutrition. I'd like to conclude by acknowledging and thanking students in my lab, Dana Lowry, Michael Roth, and Barbara Husik, for their very important roles in the data analysis and creating the infographic. I'd like to thank my collaborators at the University of Manitoba for allowing us to come to their lab to conduct this research. And I'd like to extend a warm appreciation to all of my colleagues, all of the Comet 2 co-investigators, as well as the participants of this study. Thank you very much for your attention.